Okay, class, we are now again looking at the acrosome reaction. The acrosome again is a vesicle located at the top of the sperm head. Here is the membrane, plasma membrane, also called cell membrane for the sperm, and here's the membrane for the vesicle. The vesicle again is the acrosome because it's just a regular vesicle. Okay. So what will happen is before activation, here's what it looks like. Then when this head binds to the zona pellucida around the oocytes in metaphase 2, there's fusion and exocytosis. What will happen is it will bind, then this membrane will bind an exocytosis, and so after the acrosome, you see it's not there anymore. Okay. So then when the sperm enters the oocyte, so now we're saying that it's entered the oocyte, calcium is released from the endoplasmic reticulum. Calcium is released from the endoplasmic reticulum. This calcium wave travels through the oocyte to the opposite side of the entrance of the sperm. The calcium has several effects. Okay, now the calcium enters in because it's a higher concentration, and so when the sperm enters the oocyte, calcium is released from the endoplasmic reticulum. Okay, a calcium wave travels along the oocyte, the opposite side from the entry of the sperm. The calcium has several effects. It prevents other sperm from entering the oocyte. So I'm going to come here. This calcium released from the endoplasmic reticulum. I'm going to come here. So here's a picture again of the acrosome. Acrosome releasing chemicals, proteases, hyaluronidase, dissolves, enters in. Okay, so it's going past this zona pellucida area here. So now I'm in, and now we want to prevent other sperm from entering. We don't want to have a bunch of sperm entering. Several sperm entering and fertilizing the same could cause twinning or other problems. Okay. Now this calcium also activates the oocyte to finish meiosis. Now remember, it's in meiosis 2, metaphase 2 at this point in time. Okay. Let's go a little further here. Only one sperm is allowed to penetrate under usual circumstances. Two mechanisms ensure monospermy. A fast block, the membrane depolarized presenting, that has to do with this calcium aspect, and then a slow block to polyspermy that has something to do with some zonal inhibiting proteins that destroy sperm receptors. Those receptors we talked about on there cause sperm already bound to receptors to detach. Okay, so there's a couple of things happening here in that particular, all to prevent several sperm from entering. Okay. All right, so now we go and we look at, okay, when the sperm enters. So now here we are, 12 hours after the sperm enters the oocyte, the nuclear envelope around the ovum disappears and the chromosomes join to form a diploid zygote. That's the key. The key is to get this end and for the sperm to release its genetic material. There's 23 pieces to join and form a diploid zygote. Monozygotic twins, single ovum splits. Okay. Dizygotic, 2 eggs are fertilized by 2 sperm. So in the monozygotic, the single ovum splits. Okay. But in dizygotic, 2 eggs are fertilized. Sperm contributes half, egg contributes half, as we know. Okay, let's go further. So we then go to, upon entry of the sperm, the secondary oocyte completes meiosis 2, cast out the second polar body. It's the second polar body. We already cast out the one in meiosis 1, so this is now the second polar body. The ovum nucleus swells, and the two nuclei approach each other. When fully swollen, the two nuclei are called pro-nuclei. Fertilization when pro-nuclei come together. Okay? So what we're looking at is this. Here's the situation. Following fertilization, here we are, primary oocyte, first polar body, ovulation, right here. Fertilization comes in. We now have the zygote. The sperm releases genetic material into that, and now the nuclear membrane disappearing and the continuation of the whole process to now give the one egg that is, so in other words, right here, let's go back again. Once fertilization occurs, then as far as the oocyte, it needs to complete its process of meiosis. Okay, once it does that, that means now it's one end. Now it can combine with the one end from the sperm to the one end of the egg, should we say, and now that's where we are. Okay. All right. So this is in vitro, but before I go to that, let me go back here to be sure we have everything. So here again is the same picture. Pronucleus, female pronucleus, male pronucleus. Okay. Mitotic spindle that's occurring. Completion of everything here. And then cleavage giving two daughter cells. That means mitosis. Now right here, these two need to stay together. If one separates from the other, then each one could form an entire human. Let me go back. Go back here. So if both were to activate, then that's where we could get dizygotic twins and monozygotic twins. Okay. Okay, so identical twins, single ovum, but one sperm. So right here, these would separate. Two daughter cells, and each would go its own way. So it needs to stay in that zone of pellucida. Dizygotic would be if the woman double ovulated, shall we say, which can occur. And two sperm fertilized two eggs. So these are called identical. These are non-identical. Okay. So this is what we're looking at here. All right. We'll go a little further. This is in vitro fertilization, but before I go there, let me just be sure. Okay, so the pre-embryonic period. Remember we talked about the pre-embryonic is like the first 2 weeks. The first cleavage produces two daughter cells called blastomeres. Then there's a bunch of multiplication till you get to a morula. So you would first get the zygote, the fertilized entity. Then it would go to a 2-cell stage, which you don't see here, then 4 cells, then these would multiply. And once you get about, and these are the days and everything. Okay, so let's go back. The fertilization occurred generally in the fallopian tube around the ampullary end. So the sperm traveled up. It did the capacitation that we talked about. Okay, so fertilization occurs. Now it's traveling down, trying to go into the uterus. As it travels down, it starts to multiply, but the cells stay within the zona pellucida. So in other words, these cells do not get bigger. Okay, this would get us back to the cell divisional cycle. So they don't spend much time in G1 because again, if the cells would get big, you would get clogged in the fallopian tube. something we call an ectopic pregnancy or a tubal pregnancy. So you still have this zona pellucida, so you're coming down 4 cells, then you finally get to 16, which they call a marla, okay, mulberry type of thing. Then the cells are going to start to do migration. And when they start to do migration, this right here, see the degenerating zona pellucida, this right here will be called the inner cell mass, okay? The inner cell mass, or the embryonic dish, some call it. And this is where the baby will develop from. And this is the chorionic area here. This is the blastocyst cavity. Okay, so we're coming down. All right. So now I go to this here. Okay, come back here. The first cleavage produces 2 daughter cells called blastomeres, then the marla, which is the mulberry. By the 4th or 5th day, the pre-embryo consists of 100 or so cells called the blastocyst. Okay. The blastocyst is a fluid field composed of a single layer trophoblast and inner cell mass. Okay. The trophoblast takes part in placenta formation. The inner cell mass becomes the embryonic disc where the person develops from. Again, using this PowerPoint, the first cleavage, mitotic, begins 30 to 36 hours after the fertilization. So we're using that one. We go here again. Characterized by rapid mitosis, which after 50 to 60 hours forms a ball of cells called a marla, which enters the uterus about 3 days, coming in about 3 days. Division continues and forms a hollow ball of cells called the blastocyst, which has 2 parts. The inner cell mass, which becomes the person, and the trophoblast will become the chorion. And this will assist in the development of the placenta from the aspect of the developing conceptus. Okay. So here again is a picture from this PowerPoint showing that. All right. So now we go a little further. I'll bring this down some. Okay, so bringing this one down. And now implantation. Okay, implantation. So let me come over here. Here is a picture. This is actually an electron micrograph showing this, what's happening here. Electron micrograph. Implantation. So around the 6th to 7th day after fertilization, the trophoblastic cell secrete enzymes that allows the blastocyst to eat into the endometrium, really the stratum functionalis. Okay. Tropha then proliferates and forms 2 distinct layers. It thus burrows into the stratum functionale of the endometrium with the inner cell mass of the entering first. That means it's facing more the wall of the uterus. Okay. By the 7th day to 10th day, the blastocyst is completely buried. Okay. And about 75%, if you can see here, of pregnancies do not implant and thus fail pregnancies. The woman generally does not even know she was pregnant. Okay. When the trophoblast presents and is covered over, 2 layers develop. The cytotrophoblast, cells of the inner layer of the retain their boundaries, and the syncytiotrophoblast. So I'm going to end here. We'll start right here at implantation. Okay.